To recap from last time, when a eukaryotic organism wants to get energy out of a glucose molecule, the first thing it does is converts it into 2 molecules of pyruvate using a process called glycolysis, so the sugar breaking stage. The way to get the most energy out of that glucose, the most bang for your buck, is to use oxygen, because if oxygen is present, then the organism can run its citric acid cycle with an output of NADH and FADH2, and that oxygen will allow us to turn the NADH and FADH2 into the most possible ATP by running the electron transport chain. We're going to talk about the electron transport chain in the future. For now, we want to talk about this step in the middle, the citric acid cycle. The citric acid cycle is also called the Krebs cycle. Those are synonymous terms. I usually use citric acid cycle, but Krebs cycle means the exact same thing. In order to get a molecule of pyruvate into the citric acid cycle, it first has to be converted into this molecule called acetyl-CoA, or acetyl coenzyme A. So we're going to follow the fate of a pyruvate as it is converted into acetyl-CoA and as it enters the citric acid cycle. And remember, the outputs of the citric acid cycle are a little bit of ATP, some carbon dioxide, and NADH and FADH2 molecules, which are more molecular batteries that your body can't use until it converts them into ATP using the electron transport chain.
So let's follow the fate of those pyruvates. Those pyruvate molecules were created in the cytoplasm of the cell, but now they're actually going to be transported all the way inside the mitochondria into the matrix. All of this is going to happen in the inside of the matrix of the mitochondria, which is inside the inner membrane, the innermost part of the mitochondria. The pyruvate is transported there, and now it's ready to get to work. So I'm going to follow the fate of one single pyruvate, but every glucose, this whole thing is going to happen twice because every glucose makes 2 pyruvates. So a pyruvate is 3 carbons. And once this pyruvate gets to the matrix, the first thing that happens to it is it's converted into a molecule called acetyl-CoA. What this means is that one carbon has been chopped off, and the remaining 2 carbons have been attached to a molecule called coenzyme A. And we're not going to worry about what coenzyme A really looks like. It is a coenzyme, which means that it helps enzymes to function. Some enzymes require coenzyme A to function, but for us it's just going to help shuttle things into the citric acid cycle. Acetyl-CoA, acetyl coenzyme A. During this process, 1 carbon dioxide is lost, and 1 carbon has been chopped off. And we've also charged up 1 NADH molecule. Now this acetyl-CoA, this is going to join into the citric acid cycle. So up to here we're not actually in the cycling part yet. This is going to join to a different 4-carbon molecule. This molecule is called oxaloacetate, but I'm not going to be able to fit that in, so I'll just leave it unnamed. It's a 4-carbon molecule. It joins with the acetyl-CoA, and the CoA part gets chopped off and leaves. These 2 join together. 1, 2 plus 4 is 6 carbons. This 6-carbon molecule is not glucose anymore. This is a different 6-carbon molecule. It is called citrate, which is also known as citric acid Hence, it's the citric acid cycle. I believe a water molecule also enters the process at this stage, but we're not going to worry about that water molecule. Citrate is going to go through several transformations. During the process, it's going to have 2 of its carbons chopped off and lose them in the form of carbon dioxide. So we've lost 1, 2 carbons in the form of carbon dioxide. During this process, we're also going to charge up 2 NADHs and 1 ATP. And we've created a new 4-carbon molecule. Now this 4-carbon molecule and this 4-carbon molecule, they're not identical. It's going to go through some more transformations, some more changes, and all of those changes are exergonic reactions, meaning that the cell can harvest the energy from those exergonic reactions and charge up some molecular batteries. What batteries does it charge up? It's going to charge up 1 more NADH, and it's going to charge up a molecule called FADH2. FADH2 is the last type of molecular battery. It's another high-energy molecule that is not available to most enzymes in the cell. Just like NADH is not available to most enzymes in the cell, FADH2 is not available to most enzymes, but it does have a lot of energy in it. So it's a good temporary storage for that energy. Uncharged is FAD. When it gets charged up, it becomes FADH2. All right, so what is our net total amount of energetic gain here? During glycolysis, we made 2 ATPs and 2 NADHs for every 1 glucose. During the citric acid cycle, CAC, we've made, okay, remember this is for 1 pyruvate, so all of these numbers get multiplied by 2. We've made 2 ATPs. We've also made 1, 2, 3, 4 times 2 is 8 NADHs. And we've made 2 FADH2s. All of these are high energy molecules, but only the ATP can be used by most enzymes. So we've got this problem, which is we're accumulating NADHs and FADH2s. They have a lot of energy, but they can't be used for anything. So the next part of cellular respiration is how to get the energy out of those NADHs, out of those FADH2s, and convert it into ATP. That's going to be accomplished using the electron transport chain, and that's what we're going to talk about in the next video.